Advances in Nano-Biomaterials by Abraham, Amanda, and Diego. Nanomaterials are chemical substances that are manufactured and used at small scales ranging from 1 to 100 nanometers. Due to their small size, they have unique properties that make them essential and beneficial in manufacturing. They help increase precision and make manipulating a lot easier when it comes to manufacturing biomaterials. Nanomaterials are heavily used in different industries and applications, such as healthcare, cosmetics, etc. The recent advancements have proven to show that they are the future of biomaterials due to their effectiveness and benefits they have provided to the world. A recent major discovery has been found. Back at the beginning of the pandemic in 2020, when we were all trying to stay inside and avoid contracting coronavirus, the world and researchers were all searching for answers. And the recent advancement on nanomaterials shows that these biomaterials can help fight against COVID-19 and later future pandemics, even prevent them. As we see to the image on our right, lipid nanoparticles containing mRNA are found in these COVID vaccines. The way that these nanomaterials work is they detect, capture, inactivate, or inhibit coronavirus cell entry or replication in the human host cell, preventing the infection. Both the famous Pfizer and Moderna vaccines both rely on these lipid nanoparticles to carry mRNA into cells to avoid infection in the patient. Unlike traditional therapeutics, antiviral nanomaterials target chemical and physical properties common to many types of virus. This is the main difference between the vaccines now and the vaccines of the past. These nanomaterials inside these vaccines, they're being developed to engage with the virus particles directly by disrupting them or binding to them. Nanomaterials are designed to act directly toward the infection, increase the effectiveness of the antiviral drugs, and to even trigger an immune response from the patient. Like I stated previously, these nanomaterials may offer advantages in the context of pandemic countermeasures, whereas since they don't just, their purpose isn't just to help fight against one kind of virus. In this case, they have activity that can even affect many types of virus families, and they're just formulated quicker than the traditional therapeutics. The concepts of nanomaterials have been around for quite a while now, but it first started in 1959 by a physicist, Richard Feynman, and he was at an annual American Physical Society meeting at Caltech University. He described a process in which scientists would be able to manipulate and control individual atoms and molecules. This meeting had a great impact, but it wasn't until 1981 with the invention of the scanning tunneling microscope that nanotechnology began. This microscope allowed scientists and engineers to see and manipulate individual atoms, and it's because of this invention that nanomaterials is even a thing. Like I stated previously, even though this kind of science may be new, nanomaterials have been used for many centuries. An example would be the picture right on the right hand side, where the Lycurgus cup appears to be green in reflected light, but it appears to be red, purple in transmitted light. And this is only possible due to nanomaterials.
Hello, my name is Amanda Sanchez, and we will be discussing the recent advancements in nano-biomaterials. These pertain to chitosan nanoparticles, 2D covalent organic frameworks, titanium oxide nanomaterials, and gold nanoparticles. Both the titanium oxide nanomaterials and gold nanoparticles show a promising future in photothermal therapy. But for now, we will be discussing chitosan nanoparticles and 2D covalent organic frameworks. A lesser-known, very recent advancement in nano-biomaterials is the discovery of hydrolytic stability of 2D covalent organic frameworks. or more simply, porous organic polymers. In general, 2D COFs for short have been in the limelight because of their crystallinity, ordered and tunable porous structure, and high surface area. For similar reasons, they differ from other classes of polymers in terms of their structural regularity, ordered and tunable porosity, and functionalization. They are formed by reversible condensation of the molecular building block, rendering crystallinity and biodegradability. They were first discovered in 2005, and from there, they were separated into 2D and 3D COFs that have been reported. The 3D COFs, as you can see in the right side of the slide, are more used for gas storage applications because of their high surface area. And the image on the bottom of the slide, bottom left, is a 2D COF framework, which exhibits hydrogen bonding, dipolar interactions, and aromatic stacking. 2D COFs are arranged into two-dimensionally extended organic sheets, stacked in layers that generate periodic columnar pi arrays, causing them to be suitable candidates for photoelectrical properties. So as well as drug carrying, these polymers can be used for phototherapy and theragnostic of cancer. Since the discovery of 2D COFs and nano-biomaterials, they have been used and studied in the area of phototherapy, specifically photodynamic and photothermal. For context, light-absorbing organic dyes in the presence of light can elicit cell death for photodynamic therapy. Or in terms of photothermal therapy, these dyes may generate heat when irradiated with IR light, killing cells. However, these compounds have low water solubility, instability, aggregation, and low cell permeability. So the ordered pores of nanoscale 2D COFs can be used as a platform for assisted dispersion of these dyes, enhancing therapeutic efficacy. This also uses the photoelectric property, the high loading efficiency, and sustained release of payloads, properties that come with 2D COFs. The image on the bottom right, exhibits the ordered pore and framework of a 2D COF and how it can transport dyes and collect them and kill cells, as shown in rat studies. Another application of 2D COFs since its discovery in nano-biomaterials is 2D COF-based drug delivery. They act as biocompatible spherical nanoparticles, and they exhibit direct correlation with drug loading efficiency and pore size. And because of their tunable pores and tailorable surface, they are prime targets for this type of therapy. They also exhibit stimuli responsiveness and target delivery of payloads, which are indispensable characteristics of an ideal drug carrier. In the figure below, a study had shown an amine-based 2D COF, TTI COF, synthesized via aldemine condensation, which was explored for sustained delivery of a drug, quercetin, in breast cancer cells, resulting in enhanced cell killing when cells are treated with quercetin-loaded COF, compared to that of free quercetin or only COFs. Recently, research began to develop nanosheets of 2D COFs, which will allow for even greater absorption of drug molecules and superior control over release kinetics, while also maintaining extreme thinness. Although boronic acid-based COFs pose a problem because they are highly unstable in the presence of traces of acid, alkali, alcohol, nucleophiles, and atmospheric moisture, boron is also ultra-sensitive to hydrolysis. So many different types of COFs have shown instability in the face of hydrogen or water. So the pore stability and difficulty processing it into nano-sized materials limited COF use in regenerative medicine. However, a team under Akhilesh K. Gahawar are leading research in developing a new class of porous nanoparticles that can direct stem cell differentiation. The 2D COFs engineered by the team found that the high porosity of these nanoparticles could be used to prolong delivery of drugs to human mesenchymal stem cells, cells that are used in bone regeneration. They've enhanced the hydrolytic stability by integrating 2D COFs with amphiphilic polymers, macromolecules that contain hydrophobic and hydrophilic components. As a result, this enables water dispersibility to COFs. This research provides a promising future for the use of COF nanosheets in directing tissue regeneration and drug delivery throughout the human body. This may be applicable as a tool in orthopedic surgery, treating osteoarthritis, and other degenerative bone diseases. The figure on the bottom left shows the infiltration of 2D covalent organic nanosheets through our actin cytoskeleton of human mesenchymal stem cells. The figures on the right show data from the study exhibiting treatment over time and other variables for 2D COF nanosheets and stem cells. Anyway, another advancement in the field, a very recent advancement / discovery in the field of nano-biomaterials is chitosan nanoparticles. Chitosan is a very promising polymer for nano-based drug deliveries. Chitosan is a biological derived material, a cationic polymer derived from chitin through deacetylation, and it is the second most abundant polymer after cellulose. It is commonly derived from fungi and the exoskeleton of crustaceans and insects. One of the main advantages that may come about with nanotechnology is the ability to target tumors, as seen similarly in the ability for targeted drug delivery with covalent organic frameworks. Because of its functional groups, chitosan can be easily modified with other bioactive molecules so that it can support additional properties to tissue construction. For example, the positive charges on its functional groups allow it to be one of the only positively charged natural polysaccharide, allowing its functional groups to interact with negatively charged biomolecules such as lipids, proteins, DNA, and cell receptors. The figure below shows the chemical structure of chitin and how it can go through the process of deacetylation to become chitosan. And as you can see, it's derived from crabs, shrimps, lobsters, and their exoskeletons, and also fungi. So chitosan scaffolds help to provide a natural environment for cell growth. And these scaffolds can be created through several types of methods, like solvent casting, compression molding, and freeze drying. These scaffolds can be made as membranes, hydrogels, and sponges, offering a wealth of possible tissue engineering applications. Chitosan was first discovered in 1811 in mushrooms by researcher Henry Krakonaut, and it had been used and studied as a drug delivery system, biocompatible scaffolds, hydrogels, and thin films ever since then. Since its discovery, chitosan has been used very much in biomedical applications, for instance, in biological tissue engineering, as a delivery vector, as an antimicrobial for wound healing, and as a thin film, as seen in the top right. It can be applied not only to biomedical engineering, but also for food safety purposes. It's also been used in dentistry, and for scaffolding, it's recently, but an advancement of chitosan has been chitosan gelatin composites for scaffolding, which were characterized by a highly porous structure with interconnected pores, and the mechanical properties were significantly enhanced. The study by Thien Han and their team underscores chitosan gelatin composite as a potential scaffold material for regeneration. Although, despite these advances, there's been even more recent advances that we will talk about. More specifically, chitosan nanoparticles for diagnosis and therapy are being researched because of their minimal systemic toxicity to the human body, despite their aggressive cytotoxicity against cancer cells and tumors. So far, studies have formed a drug delivery systems based on chitosan nanoparticles for preclinical and clinical studies. This would be extremely helpful because statistics have shown that oral administration of drugs for cancers is difficult due to physiological barriers in the gastrointestinal tract. Chitosan nanoparticles are most suitable for local delivery to the dermis and the mucosa because of its mucoadhesive properties and permeability enhancing actions. Many studies have shown the in vivo ability for chitosan to act as an effective drug delivery system for cancer therapy and other stuff. For instance, Li Wang and his team had done a study for the simultaneous incorporation of PTH and nanohydroxyapatite into chitosan alginate hydrogels for efficient bone regeneration. The study showed that the hydrogel containing chitosan exhibits strong biocompatibility and facilitated osteogenic differentiation of RBMSC, through the Notch signaling pathway. Another study by Gagliardi demonstrated another practical chitosan derivative in drug delivery systems that showed high drug loading capacity, biodegradability, and suitable serum stability that can be utilized in cancer diagnosis and therapy through the use of chitosan. Both these studies were completed in 2020 to 2021. Another study that is not focusing on the drug delivery for oncological treatment or for regeneration is one based on improved dressing for wound healing. The study utilized a silver metal organic framework loaded with chitosan nanoparticles and polyvinyl alcohol sodium chitosan. They were used as the upper and lower layers to prepare a bilayer composite dressing for wound healing. The PAC's lower layer, which had the polyvinyl alcohol sodium chitosan, had uniform pore size distribution, efficient water retention, swelling, water vapor permeability, biocompatibility, and not very much antibacterial activity. The upper layer of silver metal showed high antibacterial activity and low biocompatibility. But because it is the upper layer, it is not in direct contact with the skin and avoids microbial invasion. The bilayer also adheres to red blood cells and platelets to promote blood coagulation and cell proliferation. This new bilayer showed promising results with significantly accelerated wound healing and minimal inflammation. An example of the dressing bilayer is seen on the bottom left image, and on the right image is medicine nanocarriers and new therapeutic drug release systems that utilize chitosan, and shows the pros of the use of chitosan and such, and disadvantages. Also, I just wanted to include that recently chitosan has been used in the development of 3D printing ink. So a limited number of studies have been undertaken to develop chitosan as a 3D printing ink, yet studies since 2020, such as the 2020 research by Moraboza, propose a combination of cross-linked polymers, including chitosan, to create photopolymerization of biocompatible scaffolds that exhibited enhanced biocompatibility, cell proliferation, and cytocompatibility on fibroblasts. So that also shows a promising future, although probably not, it still has very much to be studied because there isn't too much information on chitosan as a hydrogel for 3D printing. On the right is on the right image on the slide is drug delivery for cancer therapy. It shows how chitosan is utilized as a carrier for drugs to combat cancerous cells and tumors. Also, the left image shows how hydrogels can be used for 3D printing scaffolds and where it can be used in the body, such as on bone tissue, skin tissue, because of its biocompatibility and such. All in all, chitosan may soon be applied to all types of tissue and skin engineering to treat wounds, cancerous tumors, degenerative bone disease, and diseases of organs such as the kidney or the intestines because of its application in drug delivery.
Hello, Professor. My name is Diego Banegas, and I will be presenting the benefits, the challenges, and some of the cons of nanobiomaterials as we know them today. So to start off, what makes nanobiomaterials special? It is simply their size. They're so small that they don't behave like regular matter as we know it. They don't follow the normal laws of physics and chemistry. Since they behave differently because of their size, they belong to what is known as the quantum realm, and some of them start displaying quantum effects, specifically in optical applications and electric applications. Those effects are very observable. Um, Nanobiomaterials, as we know them today, are considered to be the future of medicine. They are extremely compact. They are extremely adaptable. As literally something in between 1 and 100 nanometers can save your life, constantly monitor your health. Not only that, we can make sensors out of nanobiomaterials. The sensors can detect moisture, heat, and a lot of things that we could implement to even crops in order to keep our food supply safe. Another application of nanobiomaterials, it's also been put in food as silver nanoparticles because they have a naturally, uh, they can repel like bad bacteria, and like they keep everything clean. It's been used in clothing, it's been used in food. Um, nano Nanobiomaterials also would also contribute to faster recoveries. It's all about making incisions smaller, procedures less invasive, less complicated, less intervention. So with nanotechnology in general, patients will not have to go back and visit the doctor twice or three more times after their major procedure. They have an accident or anything of the sorts, they can easily get treatment fast, non-invasive, and they can go back to their homes and not have to worry about rescheduling. So it's a great benefits right now, but more to come. Challenges and cons to biomaterials. Uh, Talking about the previous chitosan nanocarriers that Amanda was talking about, their instability is affected by other factors such as pH, digestive enzymes, surface charges. Um, it is a great concept and it is very useful. However, it has various setbacks at the moment. They can also suffer from low drug encapsulation, premature release, and the the just discussed and instability. Um, One of the main challenges of biomaterials today, nanobiomaterials specifically, is the lack of knowledge on the matter. Since they are so small, so hard to manipulate, so hard to manufacture, it's even harder to research them or even know the long-term effects. Um, the research is also very expensive, and usually these types of medical devices and bio like nanomaterials do not give a fast yield and revenues. So some investors are kind of struggling in this area as well. Um, there's also been proof that uh, nanotechnology can also do DNA damage, because especially metals, nanoparticles of metals can damage human DNA. Um, like I said, there's no knowledge of the long-term side effects. It's also been stipulated that what if a what if a lot of nanoparticles are inserted into a person, and then they won't like they're all the extra particles flying around inside of the person can deposit in their tissues and their organs and their bones even, which can create even bigger problems, and then they will be impossible to remove because there's so many of them. um, there is some sort of association in the United States that controls and researches all of this. It is called the National Nanotechnology Initiative. They're currently working on regulations and innovation on applications, and they've made a lot of progress in the last few years. There's also the environmental risk. Yes, nanotechnology and nanobiomaterials can contribute to a safer environment. However, their mass production and their manufacturing can lead to waste or even nanoparticles being so small that they're going to seep through the earth and contaminate underwater, like underground water, which feeds to our drinking water, which feeds the crops of the food that we eat. So it'll pretty much be a catastrophe if something like that, like this, gets out of hand. So still a lot of progress to be made, and there's still a lot of challenges at the moment um, for nanotechnology in general, but also for nanobiomaterials specifically. So, as far as the future research, this is one of the most interesting parts of the presentation because it tells us what's going to be possible in the future, what we're looking at in the future, and why people are investigating and researching on nanotechnology in general at this current time in the world. So, first of all, there's a diagram on the slide showing a small robot-like nanostructure attacking bacteria and or pathogen, some sort of pathogen inside the body. This is not yet possible in this scale, but this would be what we would call an artificial nanostructure inside the body. This would pretty much be a device that can roam around your body. It's so small that it can literally go through your blood vessels, blood-brain barrier, hard to access places in the body. This little device will be able to detect abnormalities, detect blockages, detect any sort of inflammation, infection, and it will run and like a white blood cell, which is just an automated n- nanostructure inside our body, it would act like a super white blood cell because it can move all around your body. It can remove blockages, attack pathogens. This is one of the future applications of nanotechnology and also nanobiomaterials. Um, this can also be taken to the next level by doing what they call personalized medicine. We're all different. We all need different medicines, different ratios of medicine, different doses of medicines. And a device like this could easily just trace your entire genome and do a DNA personalized medicine like dosage and treatment, which is something that's very far off at the moment, but that would be ideal for everyone since everyone is different. Um, Another one of the major future researches to be done is in medical imaging. Medical imaging is there to detect diseases, to detect tumor, like everything inside the body that is not easy to see from the outside or diagnose can be done by medical imaging. Nanoparticles can be used as contrast agents in order to increase the resolution of the image produced by the by X-rays or anything. This can easily lead to diagnosing diseases earlier than what doctors are capable of right now. Not only that, but also treat them faster, which in most cases the survival rate go up multiplicatively just by detecting the disease earlier. Um, Other research also includes bone tissue and organ regeneration. Nanotechnology can definitely be used to for those applications. Health monitoring, we can do something called the combination product. The combination product is a future project of nanotechnology, which is pretty much something like a pill that's nano sized. That pill can monitor your health, can do combination product means a nanoparticle that can perform multiple operations, right? So it can detect, it can act, it can fix, uh, it can recruit. Um, so pretty much it's something that can not only detect it and then you have to go to the doctor and then they do a surgery or a procedure. It's an instant detection and instant treatment type situation. So you would have an army of doctors living inside your body at all times, which sounds sci-fi right now, but it is something that's on the way. Uh, Now, the last thing I'll be discussing is magnetic microsurgery. Like I said before, everything in medicine has been about making everything smaller, machines smaller, pills smaller, uh, syringes smaller, everything. So microsurgery would do what I was talking about before, the quicker recovery, less intervention. Nanotechnology pretty much helps to fix the shotgun approach of, for example, antibiotics, which don't only interact with the pathogens that you're trying to get rid of the body, but they also interact with normal cells in the body, which is a problem because you're damaging nearby tissues and cells that you're not supposed to be damaging. With nanotechnology, you can pinpoint and go straight to the issue um, without further repercussions. But however, of course, this slide, most of it to a certain extent is still theoretical and still in the process. So, in conclusion, Nano-biomaterials is extremely essential in the medical field as it relates to all of the following: pharmaceutical drugs and their preparation, conception, diseases in general, especially in cancer treatments, diagnosis, therapy, and surgery. Um, and to finalize, nano-biomaterials, another reason why it's so hard to tackle and research and perform experiments on is, well, it is an interdisciplinary field. It's not just it by itself. It depends, even a simple project needs physicists, mathematicians, engineers, chemists, biologists, and even the person that is going to be applying like the product, which is, for example, an orthopedic surgeon. So there's a lot of people, resources, and in general, a lot needed to research and perform applications with this particular field. But that was our presentation, and we hope you enjoyed it.